Imagine you are trying to build this massive, complex new wing onto your house.
Right, a huge project.
Yeah, exactly. You've got all the blueprints drawn up, the framing is going up beautifully, the drywall is, ready to be hung. But there is a huge structural problem hiding underground.
Ah, the plumbing.
You guessed it. The main water pipes supplying this entire construction site are like the size of standard garden hoses.
That is not going to work.
No, not at all. The site is absolutely starving for resources. The pressure builds up, the plumbing strains under the demand, and eventually the whole system just starts breaking down, flooding the rest of your house with debris and, well, toxic runoff.
It's a complete disaster.
It is. And usually when we talk about a medical diagnosis, there's this expectation of clinical precision, right? But when it comes to pregnancy, that chaotic garden hose scenario is the reality of one of the most dangerous, unpredictable, and really mysterious complications out there.
Preeclampsia.
Yes, preeclampsia. Welcome to the Deep Dive. We are thrilled to have you here with us today.
You know, the human body is essentially an interconnected plumbing and electrical system, and preeclampsia is, it's the ultimate biological bottleneck.
A bottleneck that affects both the mother and the baby.
Exactly. We are dealing with a situation where the demand for resources vastly outpaces the supply, and the entire system just goes into shock because of it.
So our mission today is to figure out why those garden hoses fail to expand, and more importantly, how science might finally fix them.
Which brings us to the source material.
Right. We are pulling apart a breakthrough 2026 paper from the journal Cell, Death and Disease. And the title is a bit of a mouthful. It's called Role of the Trophoblastic BMP7-TGM2 Axis in the Pathogenesis and Prevention of Preeclampsia.
It's dense, but it is fascinating.
We are going to translate that into plain English for you. Because the researchers didn't just find the microscopic root cause of this condition, they tested a potential cure using a protein that until now was primarily known for healing bone fractures.
Yeah, finding an FDA-approved therapeutic for broken bones that might actually save failing placentas is a massive shift in how we view maternal medicine.
It really blows my mind.
My goal today is to map out the microscopic chain of events that makes this possible, connecting a tiny cellular relay race to the very real clinical outcomes for mothers and babies.
I love that. But I want to start at the clinical level before we shrink down to the microscopic level.
Sure, set the stage.
What does preeclampsia look like in the real world? Like, why is it considered so devastating?
Well, clinically, preeclampsia is a major hypertensive disorder, meaning it is characterized by dangerously high blood pressure.
And it's pretty common, right?
It is. It affects roughly 3 to 5% of all pregnancies worldwide, usually presenting after 20 weeks of gestation.
So halfway through.
Yeah. A mother might go in for just a routine checkup, and suddenly her blood pressure is through the roof. It's often accompanied by protein in the urine, which points to kidney strain. And it can rapidly escalate to end organ dysfunction. We're talking about severe liver issues, acute kidney damage, and even neurological symptoms like seizures.
What always shocks me is the standard of care for this. Despite all the advancements in modern medicine, the only definitive treatment we currently have is delivering the baby and removing the placenta.
Even if the baby's premature.
Right, even if it is dangerously early in the pregnancy. That just seems so drastic.
It is drastic, but the reason for that measure is that preeclampsia is fundamentally a disease of the placenta.
Okay.
The maternal symptoms are secondary. The moment the placenta is removed from the mother's body, her high blood pressure and organ strain typically begin to resolve.
So the placenta is the culprit.
Absolutely. To understand the origin of the disease, we have to look at how a healthy placenta forms.
Okay. Take us back to the beginning.
Early in a normal pregnancy, the fetus produces specialized cells called villus cytotrophoblast, or VCTs.
Wait, break that term down for me. Villus cytotrophoblast.
Sure. Villus refers to the tiny hair-like projections on the placenta that increase surface area for nutrient exchange.
Okay, got it.
Cyto means cell, and trophoblast essentially means a cell that supplies nourishment. So these are the foundational cells of the placenta.
The building blocks.
Right. And a subset of these VCTs have a very specific, almost aggressive job. They transform into what are called extravillus trophoblasts, or EVTs.
EVTs.
Yeah, and these EVTs physically migrate away from the placenta and invade the mother's uterine lining.
So they act almost like an invasive root system. Or maybe a specialized demolition crew.
Yeah.
Because they have to dig into the mother's tissue and find her blood vessels.
That is exactly what a demolition crew would do. Their physiological purpose is to find the mother's spiral arteries, tear down the muscular walls of those narrow, high-resistance vessels.
The garden hoses.
Exactly, the garden hoses. They tear them down and completely remodel them. They rebuild them into wide, floppy, low-resistance fire hoses to ensure the growing baby gets a massive, uninterrupted blood supply.
That sounds like a highly coordinated invasion. I'm guessing in preeclampsia, the demolition crew just, goes on strike?
Basically, yes. The invasion is shallow. The EVTs stop working halfway down into the uterine lining.
So they just give up.
Because the demolition crew fails, those maternal spiral arteries are never remodeled into fire hoses. They remain narrow and stiff.
Leading to the bottleneck, the starvation of resources.
Precisely. The medical term is placental hypoperfusion, meaning inadequate blood flow. But it isn't just a low flow, it's a stuttering, fluctuating flow.
And that fluctuation is bad.
Very bad. That fluctuation causes something called ischemia-reperfusion injury.
Wait, ischemia usually means a lack of oxygen because blood flow is blocked, right?
Yeah.
What is the reperfusion part?
Reperfusion is when the blood suddenly rushes back in.
Oh. But wouldn't restoring blood flow be a good thing?
You would think so. But when tissue is starved of oxygen, a sudden blast of highly oxygenated blood actually causes severe cellular damage.
Really?
It creates a massive burst of toxic free radicals. This puts the placenta into a state of severe oxidative stress and endoplasmic reticulum stress.
That sounds intense.
It is. Basically, the protein-making factories inside the cells start misfiring and breaking down.
So the placenta is essentially suffocating and then getting battered by these bursts of oxygen.
In response to that trauma, the stressed placenta starts releasing toxic anti-angiogenic factors, meaning molecules that destroy blood vessels.
Oh, no.
Along with heavy inflammatory signals, and it dumps all of this directly into the mother's bloodstream.
So that toxic debris flooding the system, that is what triggers the mother's systemic high blood pressure and damages her kidneys and liver.
Exactly.
It's the rest of the house flooding because the pipes fail. Okay, let me pause here and just do a quick summary for you listening, just to keep this all straight.
Good idea.
Number one, preeclampsia is fundamentally a placental disorder. Number two, it happens because those fetal trophoblast cells, the demolition crew, fail to invade and widen the maternal blood vessels.
Right.
And number three, this failure starves the placenta, which then panics and poisons the mother's system. Did I get that right?
You have the exact sequence. The question that has frustrated researchers for decades is simply, why do those cellular roots fail to dig?
Which brings us to the breakthrough in this paper. The researchers decided to look closer than ever before, using a technique called single-cell RNA sequencing.
Yes. Single-cell RNA sequencing, or cRNA-seq, is a massive leap over traditional biopsy analysis.
How so?
Well, a standard tissue sample is like a mixed fruit smoothie. You can tell what it tastes like overall, but you can't measure the individual ingredients.
You just get the average flavor.
Exactly. Single-cell sequencing magically separates the smoothie back into individual strawberries, bananas, and blueberries.
That's incredible.
It lets researchers look at a placenta and see exactly which genes are turned on or off in every single individual cell.
So they compared healthy first trimester placentas with third trimester preeclamptic placentas. And they found a prime suspect hiding in those early trophoblast cells.
Bone morphogenetic protein 7.
BMP7. I know we teased this earlier, but how on earth does a bone protein end up in a placenta?
It's a fair question. BMP7 is part of a large family of cellular messengers called the TGF-beta superfamily, which generally regulates cell growth and differentiation.
Okay.
We know it best as an FDA-approved treatment to stimulate bone growth in severe fractures. But the sequencing data revealed that healthy villus trophoblasts naturally produce high levels of BMP7.
So the placenta is manufacturing its own bone growth factor.
It is.
But when they looked at the preeclamptic placentas, the BMP7 was just missing.
It was significantly downregulated. The signal was essentially turned off.
Wow.
And one crucial detail the researchers noted is that BMP7 acts purely locally within the placental microenvironment.
Meaning it stays at the construction site.
Yes. When they tested the blood serum of the mothers, they couldn't detect BMP7 at all. The problem isn't in the mother's systemic circulation, it's a localized failure at the construction site.
So the growth signal goes quiet, but biology rarely leaves an empty space. All right. When the bone protein disappeared, the sequencing showed something else taking over, an enzyme called tissue transglutaminase, or TGM2.
That's right.
Keep this simple for me. What does TGM2 actually do to a cell?
TGM2's primary biological function is to cross-link proteins. It forms incredibly tough chemical connections called isopeptide bonds between different proteins.
Like a biological superglue.
Biological superglue is the best way to visualize it. It is used in normal biology for tissue repair, cell adhesion, and stabilizing structure so they don't break down.
So it's normally helpful.
Right. For instance, TGM2 glues a specific blood pressure-related receptor, called AT1, into place on the cell surface.
Okay, I follow.
But the single-cell sequencing revealed a massive problem. In preeclamptic placentas, TGM2 is highly upregulated.
The cells are drowning in superglue.
Exactly.
So we have a missing growth signal, the BMP7, and an overflowing glue factory, the TGM2. It sounds like they have to be connected, but how did the researchers prove one was causing the other?
They took the investigation out of the human body and into the lab. They started with in vitro tests using immortalized human trophoblast cells.
What are those exactly?
These are lab-grown cells, specifically called HTR8S vino cells, that mimic the behavior of early placental cells. They also use primary trophoblasts taken directly from early human pregnancies.
And this is where they introduced the bone drug.
Exactly. They bathed these cells in recombinant BMP7 protein.
And what happened?
The cellular response was immediate. The migration and invasion capabilities of the cells skyrocketed. They started moving and digging with incredible efficiency.
The demolition crew went back to work.
They did.
The paper had a twist here that I found really clever. They didn't just watch the cells move. They sequenced their RNA after adding the BMP7.
And the data was very clear.
Yeah, it showed that TGM2, the superglue, was one of the most heavily downregulated genes. BMP7 wasn't just encouraging the cells to move, it was actively shutting down the glue factory.
To confirm that the glue was actually the thing stopping the invasion, they escalated their testing to an ex vivo model using 3D trophoblast organoids.
I love organoids. They're basically lab-grown mini organs, right? Like science fiction made real.
They really are. They are tiny three-dimensional cellular structures that function remarkably like a real first trimester placenta.
Incredible.
The researchers took these healthy mini placentas and intentionally added recombinant TGM2 protein to them.
They poured the superglue directly onto the healthy construction site.
And the TGM2 acted as a hard brake. It severely restricted a process called extravillous differentiation.
What does that mean in this context?
The specific markers we expect to see when a cell turns into a demolition crew proteins that help degrade tissue, they absolutely plummeted.
Oh, wow.
The physical outgrowth of the organoids, the actual roots themselves, shrank and stopped expanding.
So TGM2 is literally throwing down quick-drying cement on the cells. It physically restricts their movement and stops them from transforming into the invasive EVTs.
Oh, yeah.
But when BMP7 is present, it somehow sweeps away the cement, giving the green light for the trophoblast roots to keep digging.
That's the dynamic.
Okay, let me pause for our second mini recap for you listening. Number 1, BMP7 promotes cell invasion. Number 2, TGM2 stops cell invasion. And number 3, BMP7 actively turns off TGM2 to keep the invasion going.
Spot on.
But that brings up a mechanical puzzle. BMP7 is a protein floating around in the extracellular space outside the cell.
Right.
TGM2 is a gene locked deep inside the cell's nucleus. How does a signal outside the cell manage to reach all the way inside and flip a genetic switch?
It's a great question. The BMP7 protein doesn't just phase through the cell membrane and walk into the nucleus.
Right, there has to be some sort of communication chain.
There is a very specific communication chain known as the canonical SMAD pathway. I will walk us through the sequence.
Yeah, let's hear it.
It starts when the BMP7 protein binds to specific receptors on the outer surface of the trophoblast cell, known as ALK2 and ALK3.
Let me stop you at the alphabet soup there. ALK2, ALK3, SMAD. I need a way to visualize this pathway. Is it like a bucket brigade where they are passing a message along a line?
A relay race is probably the most accurate analogy. BMP7 is the starter gun or perhaps the first runner.
Okay.
The ALK2 and ALK3 receptors are the next runner, standing ready at the cell membrane.
So BMP7 passes the baton to the ALK receptors on the surface. What do they do with it?
Once they receive the signal, the ALK receptors are activated or phosphorylated. They turn around and face the inside of the cell where they pass the baton to a specific team of internal proteins.
The SMAD.
Yes, SMAD1, SMAD5, and SMAD9.
Okay, the message is now inside the cell with the SMA1, 5, and 9 team. How do they get it into the locked nucleus where the DNA is?
They need a key. Once SMAD1, 5, and 9 are activated, they bind together with a partner protein called SMAD4.
Another runner.
This newly formed complex is now capable of traveling from the outer cytoplasm, sprinting into the nucleus, binding directly to the cell's DNA, and physically blocking the transcription of the TGM2 gene.
That is a brilliant relay race.
Yeah.
BMP7 hands off to ALK2 and 3, who hand off to SMAD1, 5, and 9, who grab SMAD4, and together they run into the nucleus to shut down the glue factory.
That's the pathway.
But cells have hundreds of different pathways and relay races happening simultaneously. How did the researchers prove that this specific pathway was the one doing the work?
They proved it by systematically breaking the relay race.
How do you do that?
They used a tool called siRNAs, small interfering RNAs. You can think of siRNAs as microscopic tripwires that specifically remove certain proteins from a cell.
Okay, tripwires.
First, they used siRNAs to knock down the ALK2 and ALK3 receptors.
So they removed the runners standing at the cell membrane.
Exactly. And when they applied BMP7 to these altered cells, nothing happened. The BMP7 just bounced off, and the TGM2 glue levels stayed high.
Makes sense.
Next, they put the ALK receptors back, but they used the tripwires to remove the internal SMA1, 5, and 9 team.
So the receptors caught the baton, but there was no one inside to hand it to.
The signal died in the water. The TGM2 gene kept producing glue. They even tested a different team of internal proteins, SMA2 and SMAD3, just to be thorough. But knocking those out didn't stop BMP7 from working.
So it had to be 1, 5, and 9.
It proved definitively that the ALK23 to SMAD159 pathway is the exclusive mandatory relay race required to stop the glue.
Okay, so we have the mechanism locked in. We know exactly how it works in isolated human cells in a petri dish.
Right.
But, a dish is a highly controlled environment. Does this molecular relay race actually cure the disease in a complex living creature?
To answer that, the researchers had to induce preeclampsia in a living animal. They utilized the AD-SFLT1 mouse model.
AD-SFLT1. I'm going to need a translation for that one, too.
Of course. The researchers inject pregnant mice with a harmless adenovirus. They use the virus simply as a microscopic delivery vehicle.
A Trojan horse.
Yes. The virus is genetically modified to carry instructions that force the mouse's body to produce massive amounts of a protein called SFLT1.
And what does SFLT1 do to the pregnant mouse?
It acts as a decoy receptor. It floats around in the mother's bloodstream and physically traps the crucial growth factors that blood vessels rely on to stay healthy.
Oh, I see. So by trapping the growth factors, the decoy artificially starves the mother's blood vessels, simulating the exact vascular bottleneck we see in human preeclampsia.
It perfectly mimics the human disease. Within days, these pregnant mice develop severe high systolic blood pressure.
Just like human mothers.
The growth of the fetal pups is stunted, and the mothers suffer from glomerular endothelial injury, meaning the delicate filtering blood vessels inside their kidneys are severely damaged.
So they have created a ward of very sick mice that perfectly mirror a human mother in a preeclampsia crisis.
Exactly.
What happened when they intervened with the bone drug?
From gestational day 10 to day 13, they treated these sick mice with intravenous injections of recombinant BMP7. The clinical reversal was remarkable. The BMP7 treatment significantly lowered the mother's systolic blood pressure. It increased the weight of the fetal pups, bringing them closer to a healthy baseline.
That's amazing. But did it actually repair the tissue damage or just mask the blood pressure?
When they analyzed the tissues under a microscope, the BMP7 had physically repaired the architecture of the placenta, restoring the critical exchange interface between the mother and the fetus.
Wow.
It also healed the filtration barriers in the kidneys.
It actually reversed the organ damage in a living animal.
It did.
But, as I was reading the paper, there was a biological nuance that seemed almost contradictory.
Which part?
When they tested the whole placentas of the cured mice, the overall levels of the TGM2 glue hadn't entirely dropped back to normal. If the superglue was still there, how did the mice get better?
Well, biology is rarely a single straight line. The placenta is a highly heterogeneous organ. It is made up of dozens of different cell types from both the mother and the fetus.
Right, it's complex.
It is highly likely that BMP7 acts incredibly locally, turning off the glue only in the specific microscopic zone where the trophoblast roots need to dig.
Ah, I see.
If you grind up the entire placenta for a bulk test, that precise local drop gets drowned out by the noise of the surrounding tissue.
The smoothie problem again.
Exactly.
The bulk test masks the individual cell activity.
Furthermore, BMP7 is a multi-tool. Suppressing the TGM2 glue to help the roots dig is its primary mechanism, but it likely triggers parallel protective pathways that reduce inflammation and protect the mother's endothelial cells at the same time.
I understand it's a multi-tool, but if it is doing several things at once, how can the researchers definitively claim that the TGM2 superglue is the main villain? Like, what if the BMP7 is curing the mice through a completely different pathway?
It's a valid critique. To rule that out, the researchers needed a smoking gun. They had to prove that the glue itself was causing the disease.
Okay, how do they do that?
So they set up one final experiment. They took a new group of the sick SFLT1 mice, but this time they didn't use BMP7. They treated them with a pharmacological drug called cysteamine diHCl.
Cysteamine diHCl. What is its mechanism?
It does one highly specific thing. It directly inhibits the activity of the TGM2 enzyme.
So it chemically dissolves the biological superglue.
Exactly. They bypassed the entire BMP7 relay race and just chemically attacked the cement on the construction site.
And what were the results?
The results mirrored the BMP7 treatment. The cysteamine drug successfully lowered the blood pressure, increased the fetal weight, and improved placental efficiency.
Wow.
That was the smoking gun. It proved that hyperactive TGM2 isn't just a harmless byproduct of the disease, it is the active, destructive driver of the pathology.
So shutting down that glue, whether you do it naturally via the BMP7 relay race or artificially through a direct drug, is a highly effective way to halt preeclampsia.
Yes.
Okay, let me do our final recap to make sure you listening have got the full picture of this biological detective story. Number one, the ALK to SMAD relay race is the exact molecular mechanism that BMP7 uses to turn off the TGM2 glue. Number two, a mouse model proves that giving BMP7 can reverse preeclampsia symptoms. And number three, inhibiting TGM2 directly also works, proving this biological axis is a perfect therapeutic target.
Synthesizing all of this, the grand takeaway is that we finally have a viable, specific cellular target.
Which is huge.
For decades, the only strategy has been symptom management, giving the mother blood pressure medications just to buy the fetus a few more days of gestation before forcing an early delivery.
Right.
This research points toward a true therapeutic intervention. By restoring the BMP7 signal or finding ways to inhibit TGM2, we can actually treat the failing placenta itself and allow the pregnancy to progress naturally.
Which leaves me with a thought I find absolutely fascinating for you to ponder. BMP7 is already an FDA-approved therapeutic, right? It's formulated to heal bone fractures.
Correct.
And we know from the data that it works locally in the placenta without flooding the mother's systemic bloodstream.
Yes.
Consider where medical technology is heading. Could we be looking at a near future where we use targeted nanotechnology, perhaps microscopic lipid nanoparticles or engineered delivery vehicles, to carry these bone-healing proteins directly to the exact millimeter of failing placental tissue?
It's entirely possible.
Could we effectively cure preeclampsia at the cellular level without risking systemic side effects for the mother or the baby? It just makes you wonder what other incredible life-saving capabilities are hiding in the proteins we think we have already entirely figured out.
The biological tools to solve these massive bottlenecks already exist within us. The challenge, and really the thrill of the science, is learning how to properly deploy them.
You just have to learn how to upgrade those gardening hoses into fire hoses. Thank you for joining us on this deep dive. Keep asking questions, keep looking closely at the mechanisms around you, and never stop exploring the hidden architecture of the human body.